Go.
Okay. So, I'm happy to introduce Dian, who's going to talk about pharmacogenetics. This is really important in medicinal chemistry. When we hear about personalized medicine, we're going to explain what that is, besides, medicine that's for the person, which most medicine is. Dian hails from China, but she is hoping to take her Lawrence education and get a job in the life sciences here in the States. So we're lucky to hope to have her here. And she'd be happy to answer any questions after the talk.
Okay, so hello everyone, I'm Dian, and what I'm introducing today is the pharmacogenetics, ethnic differences in drug responses. Let's go. So let's begin with what pharmacogenetics is. Basically, it's a relatively new idea. It's subject in 1950s. So a relatively new field which studies how the genetic differences affect the drug responses. And basically, it's research on some individual responses to drugs and how variation in genes involved in mechanism. And so what does pharmacogenetics do? Basically, the goal of pharmacogenetics is to improve the drug safety and the efficacy and to reduce adverse effects. And so, in other words, to help the doctor choose the best drug for each individual patient. And let's talk about this adverse effects. There is a term called ADR, which is adverse drug reaction. It's a little bit similar to side effect, but a little bit different because side effect could be used for some beneficial effect, but adverse drug reaction is totally harmful. And in a research in 2012, there's 4.4% of all the adverse drug events that happened in a hospital stay, which means 4.4% of doctors even do not know how the adverse drug reaction will happen. And also in the same research, it reported that the cost of the adverse drug events would be as high as over $100 billion, and which means if we can kind of reduce this adverse drug reaction, we can save a lot of money. So what can we do? Basically, the best way to do that is personalizing medicine, which can be done by sequencing human genome and understand the genetic basis and learning the metabolic pathways. The last one is actually what the pharmacogenetics talks about. So that's the ridiculous, huge pathways figure about what pharmacogenetics is learning about, but that's not what we're talking about today. We're talking about just a little single sample. This drug called warfarin is a blood thinner used to treat blood clotting and other vessel and heart disease. It works by blocks the enzyme vitamin K oxidase reductase that reacted with vitamin K1, which we'll talk the whole mechanism later. And basically, this is research from 2005 and talking about the average warfarin dose requirement by ethnicity. And you can see the y-axis is the weekly dose of warfarin, and the x-axis is ethnicity. This research is about over 300 patients whose age is over 18 and has a target INR. This is basically a measurement of how smooth your blood is, this target INR, and who has warfarin management within the study clinic over 3 months. So we can see in this figure, the Asians has the least requirement of warfarin dose, and the Hispanic is a little more than Asians. Well, actually, in this research, the Hispanic people is under 20, so we're good. We won't talk about them too much. Our point is on Asian, white, and black people. And we can see white people need more warfarins, and the African American people need the most warfarins, and which means they have less risk of bleeding, because warfarin as a blood thinner, it causes people bleeding, and if you have too much warfarin, it causes you too much bleeding, which is risky. So why such differences exist by races? And the scientists found that, okay, it could be caused by metabolic pathway. And then we got this called cytochrome P450, which is the primary metabolic pathway. And the P450 enzymes are proteins of a superfamily that contains the heme group, which is structured like this. And the heme group adds oxygen to the drug molecules in phase 1 of metabolism, and which makes the drug molecule can be soluble and the body can remove them. Basically, it's located in human because this enzyme located in, well, kind of human, animals, plants, bacteria, even virus, so almost everywhere. And in human, just in human, this P450 enzyme is located in either membranes of ER or the inner membranes of mitochondria. And mostly it's located in liver because liver is the most significant issue that we use, that our body used for metabolism. And mentioned there, I used the potentially toxic compounds. Means this compound is not all, well, the regular toxic compounds we said. It includes like also like drugs because whatever drug is, if you take too much, it will be toxic to you. So I use the potentially toxic compounds, which contains all the drugs or metal ions, all the things you take in your body. There is 57 different functional CYP450 genes, which including 18 families and 45 subfamilies. You can see here are some examples of this protein, and you can see how they name them. This CYP2A13 means this protein is the 13th protein in cell family A across family 2. And this CYP2E1 means it's the first protein in the cell family E across family 2. And the last one means it's the 6th protein in subfamily A. Yep, subfamily A across family 2. So all the three are in family 2, but the first one and the last one is more kind of closer because they are in the same subfamily. And the mechanism of this P450 oxygen activation is basically it's catalyzed this reaction, which the RH is the drug molecule or the toxic molecule, and react with oxygen and 2 protons. And then after the catalysis of P450, it go away with the OH group and water molecule. So this, as I said, there's over 50 different P450 genes, and they encode over 200,000 CYP proteins, but not all of them involved in our metabolic pathways. Just some of them, and some CYP metabolites only one or just 2 or 3, just a few substrates, while others can metabolize multiple substrates, like this one we talked about, which metabolize warfarin. This one called CYP2C9, and you can know it's a nice protein in subfamily C across family 2. This one is, well, as most of P450 enzyme is oxidized of both xenobiotic and endogenous compounds, and it can metabolize multiple drugs, including warfarin and NADIS and some other drugs. So why this one causes racial differences? Because it has different genotypes. In our case, there's 2 main genotypes, like these 2 main polymorphisms, which is CYP2C2 and 3. Both of them can reduce the normal metabolic activity of the enzyme, but it says 3 is a little bit more effective than 2, but both of them works in the same way. So the research reported that the carrier of at least 1 variant copy of 2 or 3 allele will require less warfarin than the people who do not carry these alleles. And so we can see in this table here, we can see this is a frequency of CYP2C2 and 3 in different races. We can see the white people have a higher frequency of this variant, CYP2C9, but while black and Asians have rare of them. So that's kind of explain why the white people need less referring than the black people. And you can still remember the figure. But it can't explain why Asian people need even less referring than the black people. So there must be other reasons. So, well, as I said, the white people need less referring than blacks because their high frequency of variant CYP2C9, 2, and 3. Okay, so why Asians with this low frequency of CYP2C9, 2, and 3 need less referring than blacks? Another possible reason is, see the last line here, which is called VKORC1 variant, and it's very common in Asians, but rare in blacks, well, kind of 30% in white people. So this very common variant VQRC1 gene is thought to be the reason that why Asian people need less reference and have higher risk of bleeding. Here, this polymorphism called a single nucleotide polymorphism, which is SNPs. The SNP is basically a single change in a single nucleotide that happens in your DNA in specific positions. And depending on the location, it can cause a different protein product and cause a change in active of protein. And the SNP is actually very different between different races. Like, actually our skin color is depending on SNPs. So that's why African American has darker skin and white people have lighter skin. That's depending on their SNP. That's depending on specific SNPs. And here is what we're talking about today. This one called VKORC1, which is vitamin K epoxide reductase complex unit 1. And the main function of it is to make a vitamin K epoxide reductase enzyme and to reduce vitamin K epoxide. And this, as well as the T415, is also located in liver, well, mostly. And let's see this mechanism. So we can see here, this is an inactive coding factor, and it's being activated to active coding factor. That's the reason that causes your blood clot. And so it's reactive with CO2 and oxygen and this vitamin K hydroquinone. And under the catalysis of this gamma glutamyl carboxylases, which is called GGCX, and then we activate it to the active coding factor, and then with a water molecule and this vitamin K epoxide. And then here the VQRC1 appears and reduce this vitamin K epoxide to vitamin K, and then reduce it one more time to vitamin K hydroquinone again. So the vitamin K hydroquinone can be recycled and reused into the activation of these coding factors. And this is called the vitamin K circle, which if it's in a good balance, that your blood is very normal. But if it works too much, you will have too many active coding factor, which cause your blood not so smoothly. So we need warfarin here. We need the warfarin to block this VQRC1 proteins and to stop this circle or try to reduce this circle process. And so you won't have so many active coding factors. And same as the T415s, the VQRC1 also has different genotypes. What we are talking about today is this one called -1639 G2A genotypes. It's basically the A allele. This change happened in the promoter region of VQRC1 gene, and alter a transcription factor binding set and lead to a lower protein expression. So that because we have lower concentration of VQRC1 protein, we just need a little bit buffering to bind them to block this VQRC1. And so the A allele carriers require lower buffering than the G allele carriers. And here is a very interesting word map which shows the frequency of 7 types of 6 SNPs in the VQRC1 gene and sorted by regions. And we can see that in the East Asian, this orange type, which is ATT-CTG, is very common. And the rest of them are this red or crimson type, which is GTC-GCA, but almost none of other types. And we can see in Africa, there's almost no ATT-CTG, the orange type. But, well, the crimson one, the green, and the darker green, and the light blue one, but no orange type. So if you still remember, that is the variant VQRC1 gene, which is very common in Asian, but very rare in the black people. And you can see in America and in Europe that they have, well, kind of part of this orange type and, well, a lot of other types. And mentioned here, this A is the SNPs we caught, which this G changed to A position. So let's see let's see how the mechanism works. So first we have inactive coding factor, and to change it to activate it to the active coding factor, we need to oxidize the vitamin K hydrochondria here by this GGCX to a vitamin K epoxide. And then we need VKORC1 to reduce the vitamin K epoxide back to the vitamin K hydroquinone. I actually skipped the vitamin K part here, but well, that's how the circle works. And then, if we need to stop this activation, we need buffering comes here to block the VQRC1, and so that blocks the activation of coding factor. And that's how buffering works. However, we have this CYP2C9 in our body, which goes to the buffering and metabolize it, and so kind of the part of the buffering won't work because it's metabolized. And only part of buffering will try to block the VQRC1. And so under the CYP2C9 effect, that the VQRC1, the vitamin K circle and activation of coding factor won't be completely blocked, but just it reduces it works. So let's see how it works in different races. In black people, okay, here's our K circle and the activation. They have, compared to the last page, they have more expression of VQRC1 proteins, and so they need more bufferings, and they have common CYP2C9, so they won't need too many. So the CYP2C9 can metabolize part of the buffering, but they still need lots of buffering to reduce this vitamin K circle works. So that's why black people need very lot of bufferings, basically because they have a lot of VQRC1 proteins. And in white people's case, they have kind of normal VQRC1 protein expression. I don't mean black people and Asian people and abnormal, but that's just regular things. But they have this variant CYP2C9 2 and 3, And so this C9T3 can reduce the buffering requirements. And so, well, they won't need so many bufferings than the blacks, but they still need kind of bufferings to deal with the SBQ-RC1 proteins. And finally, the Asian cases, because they have very low expression of BQ-RC1 proteins, so they won't need so many bufferings, and especially the CYP2C9 will metabolize some of them, but they don't. But they still need the least buffering requirements than other research groups. So basically, what I'm talking about is the type of CYP2C9 and BQ-RC1 genotypes decide whether a person will have adverse drug reaction, and after taking the buffering. But this is just a specific example of adverse drug reaction depending on the races. Basically, scientists have already known the frequency of CYP2C9 2 or 3 allele and the BQ-RC1 variant allele, which is A allele, decide this. And so what this means, it means if I have blood clot and I go to the hospital, ask the doctor for some buffering drugs, and the doctor will predict what kind of risk I will have. Like he will tell to me like, well, you are an Asian, and well, commonly you will need less buffering doses than the black and white people, so I will give you less doses. So kind of avoid the risk of bleeding. So which means we'll kind of predict the risk of adverse drug responses by races. And what's more is we can personalize this drug and by individuals by learning the sequencing of people DNA. And also there are, well, I think about 10 papers or under 10 papers talking about other drugs which has evidence of ethnic differences, and from this AC inhibitor, and also another drug called beta blocker. But well, there are evidence shows that they have ethnic differences, but none of the research has proved what kind of mechanism they have and what genes affected their effect. So still a lot of work waiting us. And also from this kind of researches, we have possibilities for personalizing drugs, which can be done by sequencing people and learning more information of the drug metabolism. Because well, if a doctor knows what kind of genome I have and what's the drug go across the metabolic pathway, and he can completely know what kind of risk I will have after I taking this medicine. So he will just analyze this and give me the best drug for individual patients for me. So basically that is what I'm talking about. And thanks everyone. And any questions?
So you said that there was the adverse drug reaction versus how much warfarin was needed depending on the ethnicity. Is there a subset of like the type of adverse drug reaction that could happen in one ethnicity versus another, or is this all the same type of adverse drug reaction?
It's depending on drugs. Like so, it still has different types of them. I think like 2 papers talk about it, but I didn't go through them.
And then kind of follow-up question, are the same amounts of blood thinners in general, would say a different type of blood thinner drug also have the similar type of division amongst the concentrations needed for the different ethnicities, or is this specific to just warfarin?
Well, I think now it's just specific to warfarin because because as I said, this AC inhibitor and beta blockers also has ethnic differences, but they're not the drugs with possibility, and so they have different ethnic differences. And I think only warfarins has this. Well, actually, only some cardiovascular drugs have this ethnic differences, and so we still don't know more about other drugs.
So, as we live in a culture of mixed ethnicities, how does this sort of singularities that you presented here incorporate into the mixed ethnicities that actually exist in our world?
Well, let me think about it. What do you mean, like how the mixture of well, like American, the mixture of races and well, honestly, I don't know. I think that the hospital and the doctors will have some solutions, but well, I think maybe can analyze the genome by well, like if I am half blood of Asian and American, like it can be analyzed by the genome that I have well, some characteristic of Asian and some characteristic of white, and so how this interact works, well, that's very complex. But I think like that's the work of how sequencing people do.
I think in the future, this is all pointing toward this idea of personalized medicine, right? So we've used breaking down that racial group and over big numbers to get some ideas about, hey, it looks like there's some differences in warfarin and this and that. But now we can use this information to go backwards and say, all right, we've got cheap genome testing right now. I'm going to look for this gene, and that's going to give me a much better example versus just grouping people up. Okay.
Just to ask another question on that, I know that like, when you're in warfarin, you have to do a lot of testing as you do it, and so is that, if I understand that correctly, that would be to make sure that you're not getting too much or too little in case the guess was wrong based on something more superficial.
Yeah, actually, as I said before, let's go back to here. Actually, this INR is the measurement to keep to make sure that you have the correct doses of warfarin. So it's basically the measurement of how many times your blood takes to form a coat. And if you have the, well, let's say the normal INR in between 2 and 3, that means your blood is normal.
Thank you. Any other questions?
In your looking at that, have you found any examples where, outside of large research hospitals, I know that's an active thing, like at the Medical College of Wisconsin, coupled with Freighter, they have the genetics wing where they address some of these things when you're having a hard to find patient. But warfarin and blood clotting, that's been around for such a long time. Have you found instances where doctors are actively using this sort of correlation to say, okay, let's try and reduce the risk even more, and just let's run the cheap test, on these specific things? Did you find examples of an implementation anywhere?
I don't know. I think a paper, well, actually just one paper is talking about what you're saying, like run the cheaper test for the warfarin, but well, it's just one paper saying about that, so I'm not sure about it.
All right, thank you.